Jun-Jie obtained his bachelor degree in biomedical science at University Malaysia in 2007. Completed his PhD studies at University of Oxford in 2011, and currently served as a senior lecturer at Advanced Medical and Dental Institute, Universiti Sains, Malaysia. His research is primarily on stem cells biology and regenerative science, and has recently shifted his interest to explore the use of natural products in conditioning stem cells in vitro to enhance the cell functions and augment the therapeutic effect after cell transplantation. Okay, well,
Very good evening, ladies and gentlemen. I'm very glad to be invited to come and share my first ever work for my graduations 3 years ago. My work is basically on stem cell biology, and I've been working very closely for do most of the cell therapy for heart regenerations. But I bumped into one of the ophthalmologists who has been working on this corneal stem cell. She has been asking me to help in a favor to try on this honey. I started this project with a very limited interest, but the result actually stunned me a lot. So it's my pleasure to share the outcome of my project. So I would like to say it's Tualanghoney. It's one of the honey we have got and we harvested in Malaysia, and it can potentially be emerging natural remedies for regenerative medicines. Before I start my work, my introduction, I would like to introduce my university. We have basically lots of campuses situated at, which one is the, two different states of Peninsular Malaysia. One is in Penang and one is Kelantan, and this is the health campus we are talking about. But my research institute is in Penang. In Penang, we have 3 different locations. One is in the main campus in the island. It's a very, very beautiful island. And my institute is actually situated in the northern region of the Penang. That's what we call Advanced Medical and Dental Institute. Right. So let us start off with the problems that we want to solve. Lots of the corneal diseases we have been encountering. Burns, inflammation, or genetic disorders can potentially deplete stem cells in the eyes and at the niche what we call limbus. That's the reason we got most of the stem cells in the limbus. And so if you have depleted stem cell, then you will have lots of problems pertaining to your visual acuity. So the only alternative to solve this problem when you have keratitis like this, conjunctivalizations and opacifications, you lose your vision. The only alternative is to remove this region, the cornea, and then transplant it with the stem cells. Then you will be able to restore it. That we are talking in a much complex and severe conditions. We can also go out to dry eye diseases, which is a very common disease we all experience, and probably some of the eye drop can already solve the problems. But what I'm talking about is why we want to look into these diseases pertaining to autologous stem cell therapy. So stem cells has greater antioxidant capacity compared to other differentiated cells because of their nature. And under oxidative stress, stem cells can exhibit telomere shortening, induce replicate senescence. They grow much slower and reduce cell renewal capacity. This is what we do not want, because the stem cell will have to constantly proliferate and replace those damaged cells. So protecting stem cells from oxidative stress may help to promote cell survival, homing, and regeneration. To achieve a successful cell therapy, we will have to make sure the cells that we are capturing retains the stemness, retain the function, so that when And that also relies on the survival, the engraftment of those cells after transplantations. And most of the cells actually die, especially in heart therapy, when we are putting cells back to the heart. Because of the oxidative stress they experience, you actually end up very low cell number that survive in the infarct regions. And same thing goes to cornea. So you have problems with rejection, problems with engraftment, and that's what we want to actually improve the culture conditions, so make sure the cells actually survive and retain the functions. So in order to make sure the cells actually be able to tolerate or withstand with the oxidative stress, we can actually do it by maintaining a reduced environment at the site of transplantation through adjunctive therapy with dietary antioxidants. Or we can add an antioxidant supplement to cells during the in vitro expansion prior to transplantations. So all these in vitro manipulation of these cells can actually affect the therapeutic outcome later on. The efficacies of the later strategy is supported by studies showing the potentials for supplemental antioxidant in the culture medium to enhance the intracellular antioxidant activity of the stem cells, prevent cellular damage, salvage culture-induced loss of stemness. So people have actually proved it. Why we are looking at honey, we actually, if you search online, you can actually see lots of papers, lots of not so scientific evidence or testimonial saying that honey actually helps to helps in your eyes. If you have something like tiredness, dry eyes, it actually helps to some extent that we do not have any scientific evidence to support it. And so we are interested in this honey. The origins is actually from the honeycomb of the Apis dorsata, this bee, super big bee, that actually built the hive very high on top of this Tualang tree, the complex cells. It's a very huge tree in Malaysia, and it's very common in the tropical forest in Malaysia. We have lots of publications by some of our colleagues on Tualanghoney. We already shows that they have the higher phenolic content compared to other honey, and they have a very superior and repair some of your cornea to make you have a much fresher vision. So we want to actually look from stem cells perspective. So the culture stem cells, we got it from Gipco basically, and we characterize them based on the stem cell markers. And this is the cytotoxicity 3, which indicates cornea keratinocytes marker, which means if the cells express this marker, they are no longer stem cells, they are marked or differentiated keratinocytes. And this marker is actually the putative stem cells marker for cornea stem cells. And yeah, this we actually characterize it using the peripheral microscope and immunofluorescent labeling, and we confirm that they have retained their proliferative capability and express some of the stem cell markers, such as the P63. We also actually tested on different concentrations of honey and see how they actually affect the gene expressions related to its stem state. So we tested on the ABCG, the ABC ATP binding cassette transporter 2, which is also one of the site population marker for stem cells, and also the connexin 43, one of the stem cell markers, and one of the differentiated marker for keratinocytes, and K12 is cytokeratin 12 marker, also indicates the differentiation. The reason why I actually changed the terminology of human corneal epithelial progenitor cells is because of the expression of the connexin 43. They have a much higher expression of those marker, which means they have semi-committed to that lineage. And with the culture of Tualanghoney, we actually see not much of a difference in terms of their stemness, the expression of the ABCG, and very consistent of connexin 43, but at a very high concentration, you get a much reduced ABCG marker. Right, this is kind of like different from what I have read about natural products. All this while people try on natural products is want to try to kill the cancer cells. And in my projects, we want to actually find an optimal concentration to culture the cells. The ultimate aim of doing this is to keep the cells alive. And also not to actually have the natural product to influence the function and the stamina of the cells. So we try first with the viability. What concentration is the optimal one? We try a range of the concentrations and we see a much reduced viability at a very high concentration. It's not so high, but it's 3.33%. The rest they maintain comparable to the control untreated one. And that can be attributed to the pH of the honey. When you increase the concentration, you actually increase the acidity of the medium. So that may be the causative reason why they actually kill cells. So some of the past publication, if you see, they're trying to actually use raw honey and to treat eye directly using the raw 100% honey. I don't understand sometimes because it is very acidic and it can be very harmful to your eye. So it will be advisable to use a much reduced one rather than the concentrate 100% Tualanghoney. So this is the data that fascinates me. If you increase the concentration of Tualanghoney in a dose-dependent manner, you get actually rate of migration much faster, and get peaks at 0.4%, but not in the 3.33% because that concentration is actually toxic to the corneal epithelial progenitor cells, and that is significant. And we try on those hydrogen process scaffolding assays. People already prove it, and we're trying to reproduce the results, and they are comparable. So when you increase, if you have increased concentrations of the Tualanghoney, you get more of the inhibitions across various concentrations of hydrogen peroxides. So what's next? We want to know what's the concentration of hydrogen peroxide that kills the corneal epithelial progenitor cells. That's very common when you have contact lenses that uses some of the cleaning agent that contains hydrogen peroxide that can actually help be very harmful to your cornea. And we already proved that at about 50 micromolar, you actually kills almost half, slightly more than half. And at 100 micromolar, you kill most of the human corneal epithelial progenitor cells. We don't actually stop the experiment there because we want to know whether this Tualanghoney can actually give some of the benefits to these cells to go against this hydrogen peroxide. So we try different. We try at 100 micromolar of hydrogen peroxides, and we treat with a different concentration of Tualanghoney. You get much lower dead cells, at 0.4%. So we started to look into what is really in there, and this Tualanghoney is unlike the one where we are eating because I have actually filtered, processed it a little bit. It has to be a micromolecules that may be able to pass through 0.2 micrometers filters. And these are the things that we found in this Tualanghoney, that the majorities of the phytochemicals are the 5-hydroxymethyl fluorocarbohydrates. And it's one of the antioxidants that has been shown in the previous studies using GCMS. So what are the advantages of whether this phytochemical that we have identified is responsible for the observed benefits we see in corneal epithelial progenitor cells? We try on the negative control without the stress. You see a little bit of the toxicity compared to the control using the compound, the purified compound we borrowed from Sigma. And we proceed with the stress, you get much higher cells survive following the stress using 0.4% Tualanghoney, and with the control using 100 micromolar ascorbic acid. And ascorbic acid seems to be better than the Tualanghoney. But if you stress the cells with 100 micromolar hydrogen peroxide, you get a much better survival with Tualanghoney, and it's comparable with the ascorbic acids, but not using the purified compound. This actually shows that the resistance to oxidative stress is conferred by Tualanghoney. Various phytochemical compounds that's present in Tualanghoney can be the antioxidant, because we have actually shown that the antioxidant can actually confer the resistance to oxidative stress in this instance. So conclusion we can actually draw from here is the low levels of honey favors the cell's viability and gene expressions. It won't actually affect the stemness and the functions. Tualanghoney improved the migration in a dose-dependent manner, but not at a very high concentrations. Tualanghoney scavenged the hydrogen peroxide in dose-dependent manner and enhanced the cell's resistance to oxidative stress. However, further mechanistic study is required. And Tualanghoney contains non-active compound with antioxidant properties that may actually favor stem cell activity. We have already extended some of our study into looking at the cardiac stem cells and how they actually improve in terms of the functions and differentiations. We have not actually completed the projects, but we are almost there. So whether the ideas of trying to actually further down the road, doing all the mechanistic study, what actually gives the benefits, what actually makes the cells to have much of the benefits in terms of mobilizing the stem cells, repair the cornea. We want to ultimately reach this stage to turn this honey into honey-based eye drop. So with this, I acknowledge all my members in Universiti Malaysia. We have lots of collaborators to complete this work. This is the ophthalmology I talk about, who approached me to do these projects, and we have various supports from people from different universities in this project, and it's funded by a short-term grant at Advanced Medical and Dental Institute. Thank you.
Give us an interesting topic. And do you have any question? Okay, please.
So the concentration of ascorbic acid in the eye can be up to 10 millimolar.
10 millimolar.
10 millimolar.
Right.
So why do you use your 100 micromolar? Do you think it reaches the optimal benefit?
Probably the ascorbic acid can actually reach 10 millimolar in the eye. But what I'm trying to show over here is, even if with 100 micromolar of ascorbic acid, you have actually give some benefits to the corneal epithelial progenitor cells. What we are trying to compare is to make this ascorbic acid control the antioxidant properties of the ascorbic acid actually give the effects. And we didn't actually find any of the ascorbic acid in the honey, but we have lots of antioxidants. So we presume that the antioxidant properties inside the honey is responsible for this resistance to oxidative stress.
And well, let's say if you culture your cells in the action of vitamin C, that's not physiological, because in the human body there is always vitamin C about.
If you are talking about normal physiology, when it comes to cell therapy, it can be completely different. When we have the keratoplasty, when you remove those damaged cornea, it actually induces lots of oxidative stress. And we want the cells to survive with that environment before you can actually engraft and do the repair